हेलो हेलो हेलो
हेलो। हाँ रिंदम, आवाज आ रही है?
हाँ सर, आवाज़ ये सर, अब आ रही है आपकी आवाज़।
एक सेकेंड। हाँ, राधा राधा रमन जी आ चुके हैं। हेलो राधा रमन, हाउ आर यू? मोस्ट वेलकम। आज हमारे साथ हमारे स्टूडेंट्स भी हैं, मेरिट लाइफ साइंसेज के, जो CSIR की तैयारी करते हैं और रिसर्च की तैयारी कर रहे हैं, फ्यूचर रिसर्चर हैं आप ये कह सकते हैं। तो अरिंदम, गिव ए वेलकम नोट टू द सर एंड देन
यस सर। ओके
ही
सर,
विल
एवरीवन इज
स्टार्ट।
जॉइंड सर।
अभी 5 मिनट वेट कर लेते हैं क्योंकि अभी टाइम है, 3 मिनट का टाइम है।
यस सर।
हेलो।
यस सर।
हाँ जी, हाँ जी।
यस सर।
हेलो सर, कैन यू हियर
हाँ
मी?
जी, हाँ हाँ हाँ, यू आर ऑडिबल, नो प्रॉब्लम।
नमस्ते सर जी।
नमस्कार नमस्कार। बहुत
कैसे
अच्छा
हैं?
लग रहा है, बहुत दिन बाद देख करके इसी बहाने कोरोना की वजह से मुलाकात हुई, हमारे आपके बीच में बातें हो रही हैं, है ना, एक अच्छे प्लेटफॉर्म पे। कोरोना ने सबको मिला दिया ऑनलाइन।
नहीं, ये तो है, कोरोना आने से कुछ टेक्नोलॉजिकल चेंजेस हुए हैं जो मतलब काफी अच्छे रहे।
रिवॉल्यूशन आ गया, लोगों को हाइजीन के बारे में पता हो गया, सबसे बड़ी चीज है। रहना आ गया,
हाँ हाँ।
नहीं तो पूरा आदमी केयर ही नहीं करता था। ये तो एक बहुत अच्छी चीज हुई।
वो भी है, और फिर लोग फालतू का ट्रैवल करना बंद कर दिए, नहीं तो बहुत ट्रैवल ज्यादा हो रहा था एक्चुअली।
हाँ, अनावश्यक ट्रेवल बहुत हो रहा था, खर्चा बहुत आ रहा था, इसका एक चैलेंज।
खर्चा भी था सर, और टाइम बर्बाद होता है बहुत ट्रैवल करने में।
हाँ हाँ, वेंकटेश, हाउ आर यू? एनी क्वेश्चन? सो आई थिंक अभी और लोग ज्वाइन कर रहे हैं। देयर आर 30 रजिस्ट्रेशन।
ओके।
30 लोगों ने रजिस्टर किया हुआ है, मैं दिखाता हूँ, लिस्ट हमारे पास है। एंड आई डोंट नो अभी कितना टाइम लगेगा उनको, लेकिन हम लोग स्टार्ट कर देंगे। कुछ लोग लापरवाह भी तो हैं, अभी कोरोना ने ये नहीं ठीक किया लोगों को, लेट लतीफ वाला काम है ना। हिंदुस्तान में 9 बजे कहो तो लोग 12 बजे पहुंचते हैं, तो दैट इज काइंड ऑफ वेरी डिफिकल्ट। गुड आफ्टरनून विजय, गुड आफ्टरनून वेंकटेश, गुड आफ्टरनून एवरीवन, गुड आफ्टरनून श्रीलता। सो
स्क्रीन शेयर।
Arindam, just give a message in the group. All the students have not joined it, so I think once
ओके
again we
सर।
should make them remind.
ओके सर।
They should not follow the Indian Standard Time, and you can see
They should
the
follow Indian Standard Time.
इंडियन स्टैंडर्ड टाइम का मतलब 9 बजे कहा जाएगा, 10 बजे आएंगे, दैट इज
No,
ट्रेंड। ये
they
देखिए
shouldn't follow
30।
Indian
हाँ
standard habit, but they should follow the Indian Standard Time.
there are 30 registration, you can see here the long list, and I think people are joining or will join after the host will start everything. They will not lose a second, just will join and listen. That's a good part. And have you been in touch to Dr. Tripathi? डॉक्टर त्रिपाठी से मुलाकात या बातचीत होती है आपकी?
हाँ सर, अभी इधर गया था दिसंबर में, तो मुलाकात हुई थी उनसे।
हाँ, मेरी भी बात हुई थी, वो CDRI के उसके CMAP के डायरेक्टर थे ना, उस टाइम पे मेरी मुलाकात हुई थी। तो अभी कुछ दिन पहले ही मेरी उनसे टेलीफोनिक बात भी हुई। तो एवरीवन इज रिमेम्बरिंग आवर बैच, हमारा बैच बड़ा यूनिक था। जबरदस्त था। हमारे जूनियर और सीनियर सारे बहुत ही अच्छे जगह प्लेस हैं सारे, और जो जहाँ पे भी है तहलका मचाए हुए है।
अब सभी लोग अपना काम कर रहे हैं।
हाँ, कोई कंपनी में है, कोई वैक्सीन डेवलपमेंट में है, सरिता तो वहाँ पे है, लिपिन में है। तो काफी पूछते रहते हैं। मैंने नंबर भी सरिता ने मांगा था आपका, तो मेरे ख्याल से उसका मैसेज गया भी होगा।
सरिता का मैसेज नहीं आया था मेरे पास।
सरिता कांटेक्ट करेगी। सरिता को कुछ रिसर्च के लिए कुछ हेल्प चाहिए थी। सी वाज डेवलपिंग सम डायग्नोस्टिक किट बेस्ड ऑन द RNA। तो शी वाज लुकिंग फॉर योर कॉन्टैक्ट, देन आई प्रोवाइडेड हर। तो बल्कि वो पूछने लगी कि आप सारे सीनियर के संपर्क में हो। हमने कहा केवल सीनियर नहीं, जूनियर के भी संपर्क में हो। आप नाम बताओ।
नाम बताओ, किसका किसका एड्रेस चाहिए।
हाँ, किसका चाहिए। यहाँ से लेके IIT दिल्ली से लेके शिल्पी खन्ना से लेके सबका, अरुण सिकरवार से लेके सारे जुड़े हुए हैं मेरे से। और आज से नहीं, मैं तब
एक्चुअली
से ही
आजकल
जुड़ा
क्या
हूँ।
हुआ ना कि सर टाइम नहीं मिलता तो फेसबुक में चेक नहीं करता हूँ, इसलिए बहुत सारा कांटेक्ट गड़बड़ा गया मेरा।
नहीं, अच्छा है, गड़बड़ा जाए।
तो
डिस्टर्बेंस कम रहता है।
नहीं, अब धीरे धीरे काम इतना ज्यादा हो गया ना कि टाइम नहीं मिलता। फेसबुक पे उतना टाइम
बिल्कुल
रहता नहीं
बिल्कुल।
है।
हाँ जी, हाँ जी, हाँ जी, ठीक है।
हम्म,
So
तो
Venkatesh, yes, we are going to start now. Enough, there is enough wait. Now we are not going to wait. So I
Okay,
think
sir.
you can, Arindam, you can
Yes,
start,
sir.
give
Okay,
a
sir.
brief welcome speech, Dr. Saab, and then we'll start. Okay.
Okay. Good afternoon to everyone. I am Orindam Das Bakshi, MSc Microbiology student on behalf of Merit Life Science. We are looking forward to make this international webinar interactive with our chief guest and honorable speaker, great micromolecular biologist Dr. Radharaman Pandey sir, who is currently working as research and teaching associate at University of Geneva, Switzerland. And Dr. Radharaman Pandey sir has done his bachelor's from St. Andrew College, Gorakhpur, and his master's from the same college in organic chemistry. He has also done his second post-graduation in biotechnology from Banaras Hindu University, Varanasi. Dr. Pandey has done his doctorate in medicinal genetics from Uppsala University, Sweden, and his postdoc from EMBL, France. With around 22 publications in reputed journals and 11 years of research experience, Dr. Radharaman Pandey, sir, has of gene expression by non-coding RNA and RNA modifications, especially his work on piRNA and antisense RNA is commendable. Thanks to Merit Life Science and Professor Dr. Shailendra Ambert, Head of the Department, Amritsar Agriculture University. Today, the topic of this international webinar, which we are focusing, is role of RNA modification in gene expression controlling in eukaryotes, which will be honorably sought by Dr. Radharaman Pandey, sir. Thank you everyone for joining this webinar again. I hope after attending this webinar, you will find something interesting areas on which you can later on focus. With big round of applause, now I, Arindam Das Bakshi, will request our chief guest, Dr. Radharaman Pandey, sir, to enlighten us on all this topic. Thank you very much.
Very good. Again, welcome, sir.
thank you, Arindam,
So,
for introduction. Now I'm going to share my
Yes, sir,
screen.
sure.
Can I?
yes, sure.
Can you see it now?
Yes.
Yes, sir, visible.
Okay, and my voice is fine, right? It's not very
Yeah.
loud or
Yes.
very low.
No, it's a fine.
Yes, sir, it's
Okay.
audible. Quite fine.
Okay, so without wasting any more time, I would directly come to the topic. As Arindam said that I'm going to talk about role of RNA modification in gene expression, mainly in mammals. I work on mouse and other human cell lines, so my work is mainly focusing on the mammals, but I do work also sometime on Drosophila and C. elegans. So I'll be sharing two small stories just to to prove the point that how important RNA modification is in gene expression control. So you all must be aware of this basic scheme of gene expression, which is called central dogma of gene expression, where DNA can replicate to form new DNA, as well as upon transcription, it produces an RNA, which then translated into protein. And this basic scheme is very essential for cellular function. It looks very simple, but all these steps, which are starting from DNA replication to transcription to translation, they are highly regulated. Each and every step is having hundreds of different regulations. And one major regulatory mechanism is by adding modification to all these molecules. And I'm sure many of you are aware of DNA modification, specifically methylation, how it is used to control gene expression in both prokaryotes and eukaryotes. Similarly, the modification of protein, for example, phosphorylation, methylation, ubiquitinylation, and there are so many modifications which then regulate the folding of the protein or the localization of the protein or even the enzymatic activity of the proteins. And this together kind of very important for regulating the cellular function. Now, the new topic is that it's not only the DNA and protein which get modified and regulated by these modifications, but RNA is also heavily modified, and these modifications serve a very important regulatory purpose. And you'll be surprised to see this little chart, this pie chart, that there are more than 170 RNA modifications identified so far, which is more than any other modification in DNA or protein. However, most of these modifications are found in housekeeping RNA, for example, tRNA, ribosomal RNA, and other regulatory non-coding RNAs. Very few of them are present in mRNA. There are approximately 13 different RNA modifications in mRNA. Just a minute, I want to minimize this because I cannot see my slide. Yeah. Okay, so there are approximately 13 different modifications in mRNA identified so far. And I guess you all know the major modification, which is called the cap modification and the poly-A modification. And these are added, these informations are not in the DNA, they are added on top of the DNA information. And the role of poly-A and the cap is very well known, how they regulate the RNA metabolism in the cells. But there are other modifications in the RNA which were identified actually long ago. If you look at this citation, they were identified almost 45 years ago. And among these modifications, sir, I don't know why my slides are not, okay, yeah. So among these modifications, one of the modifications which is more prevalent in mRNA is the methylation. This is methylation of adenosine at N6 position. And this contributes to almost 50% of all the internal RNA modification. This is present in two different forms. One, which is part of the original cap structure, where the first nucleotide, if it is adenosine, it is modified at N6 position to make M6 AM as the first cap structure. This is also present in other adenosine in the internal position, where again the N6 position is modified, which produces M6A, which you see here or here. Yeah. So there are total 3 different methyl transferases which are known to add M6A to the RNA, to the mRNA. The first and the major one is metal 3 metal 14 heterodimer, which add M6A in a sequence motif called DRAC motif, where adenosine is modified. So this heterodimer add methylation to adenosine to convert this into M6A in the DRAC motif. The second methyl transferase, which is less known, and this is called metal 16, but it's a highly conjugate, actually it's conjugate from bacteria to human. And this add adenosine, the methylation at adenosine in this motif, which is UA, CA, GA, GAA. And it add methylation in this motif only when it is part of a stem loop structure. So on the linear RNA, metal 16 does not add any modification. whereas metal 314 add modification on a linear RNA in this sequence context. Then the third enzyme, which is called PCIF1, and it was actually recently identified, as you see here, it was identified in 2019. It adds modification to the first nucleotide of the RNA, if it is adenosine. So it converts this into methyl adenosine. Of course, the substrate is not just adenosine, but this adenosine always methylated at 2O position. So, and this is done by another set of enzyme called CMTR1 and CMTR2. And these are consistent modification. And once this is adenosine, it get a further methylation by PCIF1 to make it M6AM. So today I'll be talking mainly about these two enzymes, metal 16 and PCIF1, which we are working in the lab. But I'll just give, before I go into those two enzymes, I'll give a brief introduction also how metal 314 add modification and what does it mean for the cells. So in addition to these modifying enzyme, there are also enzymes which can erase these modifications. For example, FTO and ALKBH5, they can convert this methyl adenosine back into adenosine. Similarly, the FTO can also convert the first cap modification M6A-M into normal adenosine. So this suggests that M6A modification is not a static modification, it's a very dynamic modification, and it can be regulated depending on the response, the stimulus which cells get from the internal stimulus or the external environment. So how do we study RNA modification, and why nobody actually was interested in it? Because it was identified in 1974-75, that is almost 45 years ago, but it was not being recognized as an important modification because there was no way to uh, decide or to identify this modification or in which RNA or which RNA are really modified by this one, because there was no technique to really map them precisely onto a specific RNA. In 2012, two groups, one from US and one from Israel, they developed a technique called MIRIP, or methylation RNA immunoprecipitation. So what they did is they use an available antibody which is against the M6A. So it's a very specific antibody which recognizes M6A in an RNA. So what they did is they purified poly A plus RNA, they fragmented the RNA, and then from this fragmented RNA, they immunoprecipitated M6A modified RNA using this antibody, the anti-M6A antibody. And then they eluted these immunopurified RNA and subjected them to high throughput sequencing. One minute. Uh, 1 minute, I got something stuck here. Yeah, okay. I just want to use the laser pen actually. Yeah. So after high throughput sequencing, what you see actually is this. So when you map this sequenced RNA to the transcriptome, to all the RNA, what you see that the distribution of M6A over RNA. And what you quickly notice that most of the M6A modification is highly enriched very close to the stop codon in the mRNA. And also you see a peak near the 5' end. So this suggests that the M6A is not uniformly distributed through the mRNA, but it is localized mostly close to the stop codon and at the 5' end of the RNA. This also revealed that most of the modification is done by metal 314 because this is exactly the motif which is recognized by metal 314 to add M6 methylation at the adenosine site. So, okay, once the RNA is methylated, what happened to the RNA? How does it influence the gene expression program? So here there are a number of proteins which can recognize this modification, but among them one protein family, which is called the YTS domain protein family. They are specialized in recognizing the modified M6A in mRNA through this YTS domain, and there are 5 of them. 3 of them are cytosolic, whereas 1 is nuclear. And 1 is mainly expressed in the germ cells, which we have been working in our lab before. So how the YTS domain recognize M6A in an mRNA? So YTS domain protein, they have this YTS domain which form an aromatic cage with the help of three amino acids, which are generally the aromatic amino acids. As you see here, the tryptophan and tyrosine, they basically sandwich the M6 methyl group in the adenosine, whereas the third tyrosine residue, it provides a backbone. So in this way, they very specifically recognize methylated adenosine in an mRNA through hydrophobic interaction. And then they mediate the function of the M6A in the cells. And there are a number of functions already defined or established for how M6A can regulate the mRNA metabolism. One well-established function of M6A is that through the recruitment of CCR4 NOT complex, and this complex actually what it does, it deadenylate the RNA from 3' end and then recruit the decapping enzyme, and then that lead finally to the RNA decay in the cells. So these YTSDF1 to 3, these three proteins were shown to interact with this deadenylation and decapping machinery to regulate the mRNA stability. Of course, they were also shown in different biological contexts that they can regulate the translation of M6A modified RNA. The nuclear M6A reader YTSDC1 regulate mainly the RNA splicing, RNA export, and recent paper also suggests that it can regulate the transcription itself. So this one is a very diverse protein which can regulate multiple aspects of RNA metabolism in the cells. The other YTS reader, the YTS domain protein, the one which is mainly expressed in germ cells, we have shown that this regulates the RNA stability in germ cells and very critical for the meiotic cell cycle when the germ cells form the haploid cells. So one of the molecule which is very important for all this methylation event in the cell is SAM, called S-adenosylmethionine. And recently it was shown that actually SAM itself is regulated by methylation event. So SAM is formed by the action of the SAM synthetase enzyme from methionine and ATP, which then ligate the ATP to methionine to produce S-adenosylmethionine. And this S-adenosylmethionine or SAM then used by different metals or methylation enzyme to convert DNA, protein, and RNA into methylated DNA, protein, and RNA. And therefore, the regulation of the SAM level in the cells is very critical. If you have more SAM, this can lead to higher methylation of DNA, RNA, and protein, and that can completely change the gene expression program. Whereas the low level of SAM can then downregulate the overall methylation of the cellular transcriptome, proteome, and genome, and that can have another kind of impact on the gene expression pattern. So how the SAM level is regulated in the cells? In 2016, actually, a paper from a group in US, which found that metal 16 actually regulate the splicing of the SAM synthetase gene, SAM synthetase transcript. So as I told you before, that metal 16 recognize the adenosine in a stem loop structure, and then it can methylate the RNA. It has multiple domain. The N terminus has RNA binding domain, whereas in the middle you have the methyl transferase domain. And then also there are two other conjugate domain called vertebrate conjugate region 1 and 2. And what the group showed that this methyl transferase actually bind to the stem loop structure in the SAM synthetase transcript, and this actually lead to the recruitment of splicing machinery, and which kind of create a correct splicing of SAM synthetase gene, which then lead to production of SAM in the cells. But when the SAM level goes higher, when it reach above a threshold, then what happened that since metal 16 can methylate adenosine, and if the SAM is more abundant, it can methylate adenosine quickly. And this methylation will actually just kick out the methyl transferase from the SAM synthetase transcript. And because of this one, then the splicing machinery cannot be recruited anymore, which will lead to unspliced SAM synthetase transcript, which is then degraded by the nonsense mediated decay machinery in the cells. So this way, metal 16 regulate the proper SAM level in the cells. So what we were doing then, and we actually also showed that if you mutate the metal 16 in mouse, so if you create a mouse which doesn't have a metal 16 gene, it dies very early. Actually, what we found that at 16 cell stage, we already start seeing the defect, and by the day 3.5 of the embryogenesis, all the embryos slowly start absorbed and they are dead. So when we analyze the gene expression pattern at the 16 cell stage, that is around 2.5 days of embryonic development, what we found that it was mainly the genes which was downregulated was MAT2A, that is the SAM synthetase gene. And also there was another gene which we found downregulated was the snRNA gene, suggesting that metal 16 mainly regulate the expression of MAT2A during mouse embryonic development. And what happens if you don't have SAM synthetase gene, actually, if you just wait another days, that is when the embryo reaches 3.5 days, here we see only 2 or 3 genes dysregulated. Here we see like thousands of genes dysregulated. And that suggests the power of methylation in regulating gene expression control. So when you don't have enough SAM, what can happen to the cells? However, metal 16 is although metal 16 is highly conserved from E. coli to human, some regions of the metal 16 are found only in the vertebrates, which is called the vertebrate control region, VCR1 and 2. So the protein doesn't have this VCR region in the invertebrates and in E. coli. And actually the paper in 2016, it suggested that metal 16 regulates SAM synthetase gene expression through VCR region. So this is very critical for the SAM synthetase gene expression. So then we question how then the metal 16 gene in the lower organism, for example, invertebrate, regulate the SAM synthetase gene expression, since it doesn't have the VCR region. And in fact, there was a published a mutant line from C. elegans. This was not lethal for the C. elegans, whereas in mouse, the loss of metal 16 is lethal. So this suggests that there is something different in the invertebrates which give a different way of regulation of the SAM synthetase gene. So when we analyze the methylation of C. elegans RNA, so we started what? Sorry. Any question?
No question, please go ahead. No problem.
Sir, there is any question?
Venkatesh, have you any question?
No, sir. No, sir. Sorry, sir. Sorry, sir.
Just muted. Thank you.
Okay. So the first thing we analyze in the C. elegans is what is the methylation status. So just before I go into the data, I would like to tell you that in C. elegans there is no metal 314. So the major methyl transferase in C. elegans is metal 16, and this perhaps does all the methylation. So when we analyze the methylated RNA in C. elegans, what we found that there are few genes which lost methylation upon knockout of metal 16. And one of the gene was U6SNRNA, which you see here, and also the SAM3 and SAMS4. And these are the SAM synthetase genes. So upon metal 16 loss, actually the major down regulation of methylation are found on SAM synthetase gene as well as snRNA gene. However, when we looked at the RNA expression, we were surprised that in the knockout, actually the SAM synthetase level was upregulated. And this is completely opposite to what we saw in the mouse. Mouse, if you don't have metal 16, SAM synthetase gene expression is highly downregulated, whereas in C. elegans, it goes up. So this kind of surprised us that what's going on here? I mean, you lose metal 16 and you have a better expression of SAM synthetase. So this tells that perhaps there is another way of regulating SAM synthetase gene in worms. So then we looked into the methylated RNA data into much more detail to find out where this methylation exactly happening in the SAM synthetase gene. And based on the analysis, what we found that the methylation was mostly enriched near the 3' splice site of the SAM synthetase gene. So this one is the one which is the IP from wild type, And in wild type, you do see some methylation of this 3' splice site, adenosine. Whereas in the knockout, of course, metal 16 is not there, so this site is totally unmethylated. So what can happen when you have the methylation at the 3' splice site? And this is something which we explored. So if you remember some basics of splicing, so splicing happen in this way, where you have the 5' splice site, which is generally GU, and this is recognized by U1 snRNP, whereas the 3' splice site, which is generally AG, this first recognized by two protein is called the 65-35 complex. So this is U2AF 65 and U2AF 35. So this protein recognized the 3' splice site. And this recognition then lead to recruitment of many other protein, which you see in this one. And this finally lead to the exclusion of the intron and rejoining of exon 1 with exon 2, or yeah, the two exons. So what happened if this adenosine is methylated? What problem it can have? So first we did is we expressed the U2F35 and 65, and we wanted to check if we have an RNA methylated at adenosine, whether this protein can bind to these sequences or not, because that's the major function of U2F35, 65, to recognize 3' splice site and then recruit the other splicing factor to mediate splicing. And to our surprise, what we see that in the unmethylated form, what you can see is the UTF35 and 65 bind very nicely with a very low dissociation constant between the protein and the RNA. But when we methylated the RNA at adenosine, this AG context, there is almost no binding of UTF35, 65. So what it suggests that actually the methylation of adenosine in this 3' splice site basically remove the binding of UTF35, 65, and therefore it can basically prevent all the splicing event which takes place after the binding of UTF35, 65. And this we further proved by in vitro methylation assay. So here is the splicing construct where you have exon 1 and exon 2, and this is a little intron. And what we see that if this is not methylated, you can see very clearly when you add the splicing extract, after 60 minutes or 120 minutes, you can see the splice product here. But when you have the methylation at this position, this adenosine, the splicing doesn't happen in this in vitro splicing assay. So this clearly suggests that if the adenosine is methylated, it cannot be spliced. And this actually explains why in wild type there is low SAM expression, SAM synthetic expression, and the mutant is higher, because in the wild type there is a metal 16 which can methylate the 3' splice site. So in a high abundance of SAM, actually this can methylate the 3' splice site, and therefore it can stop splicing and therefore lead to degradation of SAM synthetase. Whereas when there is no metal 16 or there is very low SAM, it cannot methylate, and that can lead to more efficient splicing and production of SAM synthetase. So this is a summary of the story from mouse, where the metal 16 actively recruit the splicing machinery. So it is important for the splicing of SAM synthetase. And when it is not there, you basically do not have the splicing of SAM synthetase, and it goes down. Whereas in worm, this regulates the splicing in a different way. It modified the 3' splice site. So when there is low SAM level, there is no methylation of the 3' splice site, and then there is a perfect splicing and production of SAM. When the SAM level is high, what happens is that metal 16 can methylate the 3' splice site, and this methylation actually inhibit the binding of splicing machinery, therefore lead to unspliced variant and finally degradation of SAM synthetic gene. So I'll take a little pause here, and I would like to see if there is any question or things are not clear, so that then we can move to the second part of the talk. Is there any question?
Yes, sir, I have a question. Yes, sir, I
Yeah.
have a question. Yeah.
Can you speak a
Sir,
bit louder?
now, sir,
I'm not.
Arindam, your voice is very low.
Hello. Yes, sir, I
Yeah.
am
Check
audible.
your voice.
Yes, sir. Yes, sir. My question is, sir, that when, yeah, so when this modification is arised in the 3' OH site at that
Uh-huh.
at that region, So
Uh-huh.
if any gene which is near to that same synthetic gene, hello sir, am I audible, sir?
Yeah,
Yes,
yeah, yeah.
yes.
Yeah, yeah. If sir, if any gene or any kind of reporter gene or any sir bio any marker gene which is located near that same synthetic gene, and if
Uh-huh.
that splicing is occurring due to that, that's the modification is occurring due to that MET-L16. So if any sir any kind of alternation can arise sir in that nearby genes, or if it get mutated once, so matlab, if it
Uh,
get mutated at that nearby gene, so so
can you,
so that
what do
it
you
will
mean by
affect the
nearby?
hello.
Yeah,
Yeah. Yes, yes, sir. I'm audible, sir. Actually, your
yeah, yeah.
voice is
You
not
you are
that
audible.
clear.
Can you rephrase your question because I didn't get what do you mean by nearby genes?
Yes, nearby genes means, sir, that the genes which are present near to that same synthesis coding genes.
Uh-huh. Okay.
I'm highlighting those genes. If
Mhm.
those genes get modified due to the alternations which are occurring in those the introns regions, in the splicing regions of those same synthesis, so those the alternation can also change the morphological behavior and of that particular organism also, and
No,
it may
actually, I mean, when you say nearby, what you mean within the SAM synthetic gene or outside the SAM synthetic gene?
no sir, within that same synthesis genes.
Well, at least in the case which we investigated, there is no other gene which can actually do anything like this. So there is no other
Okay,
gene
sir.
or transcript produced from the SAM synthetase locus other than SAM synthetase itself.
Okay, sir.
Is that the answer for your question, or you still have something?
Okay, sir. That's the answer, sir. I
Okay,
get it.
so yeah, there is no other gene inside the SAM synthetase gene. So it's basically SAM synthetase. And when it is transcribed, if there is a lot of SAM in worm, it gets methylated. And this methylation, and just to make sure that this methylation is happening at the RNA, not at the DNA level.
Yes, sir.
Yeah. And
Yes,
this
sir.
methylation then kind of prevents splicing. Whereas in case of mouse, metal 16 directly recruit the splicing machinery. So the regulation is completely opposite. So in mouse, the metal 16 is needed for splicing, whereas in worm, metal 16 activity actually inhibit splicing.
Okay, sir.
Yeah.
Okay, sir. I got it, sir.
Yeah. Any other question?
No, sir. I don't know.
Anyone, any participant want to ask any question? I think some people have joined late, and very few people are getting the concept right. That's why they are not asking questions. So RNA word is new, and it's very exciting because corona is itself not DNA. It's having RNA as a genome. So understanding this splicing mechanism give you a lot of understanding about the RNA word and RNA
Well,
mechanism. How
this is just
uh,
one thing how a little methylation event actually can regulate the whole embryonic
expression,
development,
yes. Or
right?
embryonic expression, right? So we are talking especially about the control
Yeah.
expression of by the RNA silencing mechanism, and then some methylation pattern and some regulatory like molecules like MET-LL16, right?
Yeah.
So if
Yeah.
you all of you have any question, please, you can leave.
No,
Excuse
if you
me,
have
sir.
any question, don't hesitate to ask, please.
Because discussion give you the new idea. Who
Yeah,
is
exactly.
speaking? Can you
I
introduce yourself?
can go through the
Sir,
slide again. If you don't understand anything, I can go through it again because
Ah,
I'm here just to explain. Even if you get few lines from my talk, I think that's enough for me.
yes, yes. No, no, they will get
yeah.
100%. Don't worry. We are professor. Will explain everything. Yes, please. Who is there to ask question? Please
Sir,
introduce
I'm
yourself.
Niritika.
Okay,
Uh-huh.
your name is Arindam. Change your name.
I'm okay.
Just ask Naritika,
Uh,
whatever the question is there.
so the thing is that from the top, what I understood, does the metal 16 have a different role in different organisms? I mean,
No,
in one it is
it has
in.
basically what it looks like so far, that in vertebrates it has a different way of regulating the SAM synthetase, whereas in vertebrate it regulates differently.
Okay,
Ultimately, it regulates the SAM synthetase gene expression in both invertebrates and vertebrates, right?
okay.
It's just the
The,
way it regulates is quite different.
okay, okay, sir. That's what I wanted to ask. So it need a specific study in different organisms, right? Like,
Yeah, no, because when we were looking at the SAM synthetase gene, what we realized that in vertebrate it has this specific region called vertebrate control region,
okay,
and these are important for recruiting the splicing protein to splice SAM synthetase gene. Whereas
okay.
this region was completely missing in the invertebrate, so that kind of gave us a opportunity to investigate the function of metal 16 in invertebrate and how it regulates the SAM synthetase expression in invertebrates.
Okay, sir.
And that's why we went into this area, and we realized that actually it regulates in a completely different manner.
Yeah.
Yeah.
Yes, sir.
But the
And
ultimate goal doesn't change.
yeah.
It just kind of senses the amount of SAM present in the system, and based on that, it responds. So if there are more SAM, it methylate in worms, and then it inhibit the splicing so that there is no more SAM synthetase produced and no more SAM is produced. Yeah. When the SAM level is low, it cannot methylate. So then the SAM synthetase gene expression is
Is yeah,
going
it's
on very
happening.
well, and that produces more SAM. Yeah.
Yes, sir. Yeah, got it. Thank you.
So it basically maintaining the homeostasis of SAM depending on the signal which it gets from the cells.
Yes.
Yeah. So then I'll talk about the other methyltransferase called PCIF1. And this specifically methylates the first nucleotide of an mRNA and also snRNA. So basically it interacts with the RNA polymerase 2 through this C-terminal repeats the serine phosphorylation. Actually this help in interaction of PCIF1 with the RNA polymerase 2, and this interaction actually lead to modification of the first nucleotide, and this happen only when the first nucleotide is adenosine. Yeah. And this cartoon, what it shows that there are many modifications which are happening at the same time at mRNA. So first you get the normal cap, M7G cap on the mRNA by RNA guanylate transferases. And then sugar is methylated by cap methyltransferase 1 and after that 2. And then comes PCIF1, which then methylate the adenosine at N6 position to convert into N6 methyl adenosine. So it basically methylate all the transcript which are transcribed by RNA polymerase 2, including SM class of snRNA, long non-coding RNA, as well as mRNA. And if you look at the conservation of this enzyme, it is always having this methyltransferase domain and then the WW domain, which help its interaction with the RNA polymerase 2. However, the interesting thing is that in insect, the protein is mutated in a way that it cannot really act as a methyltransferase. So that means that in invertebrates, basically in insect, the adenosine at the first nucleotide is not methylated. And this we confirm by doing RNA mass spectrometry of the RNA isolated from worm, insect, and mouse. And you can see clearly that mouse has M6AM. That means this nucleotide is modified, whereas in worm and insect, there is no such modification. The other modification, that is M6A in the other site, other than the cap and the sugar modification, is not impacted. So these modifications are always there, but M6AM is found mainly in the vertebrates. So first thing we checked is how PCF1 expression is in the different mouse tissue. So first we create a tag line of mouse where we tag the PCF1 protein with the 3-time flag. And then we did the Western blot, because at that time we were lacking the antibody for the PCF1 gene. And what we found that this protein is expressed almost in every tissue we examined, except salivary gland, as you can see the signal from the Western blot. Then we thought it might be very important enzyme, although it is not present in the invertebrate, but still it could be very important because it is expressed almost everywhere. So we created a mouse line where we deleted the methyltransferase region in the gene. So that created a knockout mouse line. As you can see from here, that in the mutant, we tested a bunch of tissue, and what we see that there is no PCF1 expression in the mutant line. Similarly, when we examine the RNA modification in this line, what we see that M6AM is completely gone in the mutant, whereas in the wild type, you can see it. And this did not impact the M6A at other position in the mRNA. But the funny thing was that this mouse was viable and it did not have any developmental defect, which kind of surprised us that a protein which is so ubiquitously and well expressed in all the tissue, it doesn't really have any impact. So one function for the M6AM was suggested that it regulate the biogenesis of snRNA in humans and mouse. So that is the first thing we wanted to check whether it really impact snRNA biogenesis. So what we did is we did a northern blotting to test if there is any change in the expression of snRNA, and we checked here U1, U2, U4, U5. These are the ones which are mainly transcribed by RNA polymerase II. As you can see here that in the knockout and wild type, there is basically no difference in the expression, suggesting that it is not important for snRNA biogenesis. And this is a quantification of the same data, suggests that there is basically no changes in the RNA stability or RNA expression of snRNA. So then we wanted to see where this modification is, whether PCF1 add modification only to this, the first nucleotide in the cap, or it also adds somewhere else. So here we devised a technique where what we did is we collected poly A plus RNA, we fragmented this RNA and degraded the RNA which are not having cap. And this can be done by an exonuclease which recognizes the 5' phosphate. So we can basically get rid of all this nucleotide, and then we can enrich the RNA with the 5' end fragments. And then we digested this RNA with two different nucleases. So the nuclease P1 can cleave all the phosphodiester bond in RNA except the bond between M7G cap and the first nucleotide. So this will not digest this bond. Whereas when we use a combination of multiple enzymes, we can cleave every nucleotide from the RNA. And then we subjected this to mass spectrometry. So what you should see here is that these bars represent the RNA modification present only in the body of the RNA, not in the cap, because this was digested by the nuclease P1, so that the modification in the cap will not be recognized by the mass spec. Whereas the blue bar represent the modification present everywhere. So if you see something in the body as well as the cap, that means that modification is everywhere. Whereas if you do not see something in the body like this one, and you see only in the full modification, that means it is present only in the cap. And with this technique, we showed that M6AM is mainly found in the cap. When I say M6AM, basically I'm talking about the M6A in the first adenosine of the mRNA, the first nucleotide of the mRNA. So M6AM is mainly found in the cap, not elsewhere. Whereas the M6A and other modifications, they are found both in the over the body, but not in the cap, basically, yeah. And the M7G, as you can see, that the experiment worked very well because we detect this only in the cap as well. So what this experiment suggests that the PCF1 add M6A to the first nucleotide of mRNA, not elsewhere in the RNA. And then we analyze the gene expression pattern in 3 different tissues, which are representative of 3 different cell types. So we analyzed in brain, spleen, and testis. So this is testis, brain, and spleen. Yeah, and what we see that there are a number of genes up and down regulated in this one, and this is a volcano plot where you see the left side of the gene, they are down regulated one, and the right side one from this line are regulated genes in all these 3 cases. But the genes which seems to be dysregulated in the mutant are mainly the predicted genes and the pseudogenes, which generally nobody study actually. And what this graph shows that although the the total proportion of predicted genes and pseudogenes in the transcriptome is very little. So they represent hardly 5% of all the genes. But when you look at the genes which are misregulated in this tissue, the predicted genes and pseudogenes, they represent a much higher proportion. Whereas in the brain, spleen and brain, they represent very tiny fraction. So then we focus our study on the testis RNA. So the first thing we wanted to analyze is whether there is any correlation between the gene expression and the loss of M6A in the first nucleotide. So we divided the gene based on their first nucleotide. So we can group them into 4 different categories. If they start with A, C, G, or U. So if the first nucleotide is A, C, G, or U, and then we wanted to see if there is any difference in the gene expression depending on which nucleotide they start with. Since PCF1 add modification only to adenosine, what we are expecting that the genes will be affected, the only genes which will be affected are the one starting with the adenosine. And actually we did not see in the spleen and brain there is any difference in the gene expression pattern. So suggesting that this methylation is not really important for the gene expression in the spleen and brain. But in the testis genes, actually what we saw that the gene is starting with the adenosine, they are more downregulated compared to the other genes. And this indicates that perhaps the M6AM again regulate the stability of the genes if they start with A. So if this is modified, they are more stable, and if it is not modified, they are less stable. So this is a bit more detailed analysis of the genes expressed in the testis. So all expressed gene, we see that the gene is starting with the A. They are much more unstable in the knockout. That is when the methylation is not there. And if we divide these genes into highly expressed gene and low expressed gene, what we see that the genes which are highly expressed, they are the one which are more destabilized in this tissue. Whereas the low expressed gene, they are not impacted by the adenosine methylation. And then we wanted to see if these are the genes which are really methylated by PCIF1. So we did a mapping of M6A on those genes. And what we see that the genes which are methylated, really, those are the ones which get downregulated in the testis tissue. So this confirmed that PCIF1 somehow has the stability impact on the mRNA expression. So if it is there, it stabilizes the RNA, whereas in the absence of this methylation, the gene get destabilized and downregulated. And this fits very well with the previously published study where what they found that what they did actually, they synthesized RNA with adenosine as the first nucleotide, but then this adenosine has different modifications. So in the first case, there was no modification. And in the second one, you have M6A modification. The third one has both the sugar modification as well as the base modification that is called M6AM. And the fourth one has only the sugar modification. And what they were testing is if there is any difference in the stability of this transcript towards decapping enzyme DCP2, which is the major decapping enzyme and is involved in the RNA degradation in the cells. And what they found is that when the adenosine was modified at the base position, that is M6A, or in this one, you see that there is a very little degradation of this mRNA. So suggesting that if you have the adenosine modified, this prevent from the decapping activity of DCP2. And that is something explain why we see genes downregulated when they start with adenosine in the mutant mouse. So as I said that there was basically no visible phenotype in the mouse when we mutated PCIF1. But when we measured the weight of these mouse, because this is something I noticed while breeding the mouse, that the PCIF1 mutant mouse, they are generally smaller compared to the rest of the mouse. So we calculated the weight of these mouse. And when we plot the weight, what you see that PCIF1 mutant mouse, they are always having lower weight, whether they are male or female, compared to the wild type or heterozygous. They have a relatively higher weight. So this is something we found very interesting, and we wanted to further investigate that why these mouse have low body weight. So the first thing we did is we went for the micro CT scanning of the whole mouse to see if there is a difference in the fat accumulation in the mutant mouse compared with the wild type, because fat is something what create more weight in the body. And what you see here in this scan is that this is the wild type mouse, which is full of fat, whereas the mutant mouse, they have relatively low fat accumulation. And when we quantify this fat, what we see that although there is not a huge difference in the body weight, we do see a very significant difference in the total fat compared to the body weight. The mutant mouse have low fat compared to the wild type. And
Yeah.
because of this, the mutant mouse have more lean mass. Lean mass is something the body weight minus fat. And we found actually that both the brown, the white fat as well as the brown fat, they are less in the mutant mouse. So this was something very interesting for us, and we wanted to further investigate and how they have low fat. So the first thing what we did is we made a histology section of the white and brown adipose tissue to see whether there is any problem in the fat deposition in this tissue. And these are the main tissue which deposit fat in the white adipose tissue and the brown adipose tissue. And you can clearly notice that in the mutant mouse, although there is no difference in cell number of the white adipocyte in the mutant mouse, but their size is relatively small, and that suggests that there is a less fat accumulation in these cells. So what it tells us that the mutant mouse have lower body weight, not because of having low number of adipocytes, but it's basically because they cannot accumulate more lipid or more fat in those cells. And the similar thing we also found in the brown adipose tissue. As you can see, I mean, there are less number of these white droplets in the white, brown adipose tissue compared to the wild type where you have multiple lipid droplets. So this experiment gave us hint that perhaps there is a defect in accumulating fat in the body, and this can of course be because of the several reasons. That is, either they can have a problem with eating food, they will be, or they can be much more active in their they can be physically more active, or they are using different way of burning fat. So this is a long experiment we performed. Is called the physiological and metabolic profiling, where we first measure the food intake over 5 days period of this for this mouse. And the dark represent the active time for the mouse, is something like the day for them, night for us. So that's time when they are more active. And what you see that there is basically no difference between the wild type and mutant mouse. So both are eating actually same amount of food during the active time as well as during the night time. This is the average of the 24 hour. And over 5 days we measured, and we did not see any different pattern of eating behavior. So they basically eat same amount of food and still they are lean. So that's very interesting. Then we also check the physical activity, that is how many times they jump on a running wheel. And this tells their physical activity. And again, here we do not see over 5 days period, we do not really see a significant difference in the wild type as well as the mutant mouse. So both are equally physically active, and still they accumulate less fat. So that is something. We further did another experiment. And here what we check is the oxygen consumption and CO2 release. And based on this one, you can tell whether they burn more fat for the energy or more carbohydrate and protein for the energy. And this is something, this might explain why they have less fat if they are burning more fat compared to the wild type. So we calculate called respiratory exchange ratio, that is the ratio between the volume of CO2 released and the volume of oxygen consumed. And if it is 1, that means they are basically using carbohydrate, because the carbohydrate burning will give equal amount of CO2 as the O2 consumed. If they are burning more fat, then the ratio will be close to 0.7. So what we see here, the mutant mouse, the wild type mouse, they are burning more fat during the sleep time and more carbohydrate during the active time. Whereas the mutant one, it has exactly same pattern during the active time, but during sleeping, it is burning actually even less fat. And this goes against what we see, that they have low fat. So they have low fat not because they are burning more fat, but it's basically they are burning less fat. So there is something else to find out why they have low fat. So we are still working on this project and analyzing the RNA-seq data to see if there is anything we can find why these mouse have overall low fat. But all these experiments actually indicate that they might have a problem in synthesizing fat, perhaps, which can lead to low accumulation of the fat in these tissues. So just to summarize how PCF1 works. So PCF1 recognize the serine 5 phosphorylation at the N terminal of RNA polymerase 2, no, C terminal of RNA polymerase 2, and then it modifies the adenosine. And this modification is important for regulating the stability of RNA in the cells. And one of the impact of loss of this modification is the reduction in the body weight of mouse, which we see in our experiment. So with this one, I would like to stop and thanks my supervisor, Ramesh Pillai, and my colleagues who contributed to these two projects, and also the different facility which are used for doing RNA sequencing, mass spectrometry, and many other things. And I would be happy to have any question if you have.
Participants?
Yes, sir, I have a question, sir.
Yeah.
Yes, sir. Yeah, sir. Have you done the lipid profile of the mouse cell membrane, sir? Mouse cells, mouse somatic cells, sir.
That's a
If you,
very good
sir,
question, actually. That is something we are planning at the moment. So we want to analyze the lipid profile of different tissues. Actually, we want to analyze the white adipose and brown adipose to really pinpoint which kind of lipid are missing in these mouse. And that can tell us what exactly going on in the mouse in the absence of this modification.
okay, sir. And sir, I think, sir, due to, sir, low fat count in the mutant mouse, I
Mhm.
think, sir, they are less tolerable in winters. I think if you, I think if you have tested,
That is a
they
possibility.
have.
That is a possibility, but that is the consequence which they can have. So we are basically interested more in the cause of this problem. Because of course, if you have a less fat, you are more susceptible in winter. So if you give a cold shock, they will be in trouble.
Yes, sir. There's the question.
Yeah. So our goal is mainly to find out why and how this modification can specifically regulate the lipid metabolism in the body. And this is something very important because, I mean, the diet-induced obesity is there everywhere. Almost 10 to 13% of the population is obese.
Yes.
So if we can find a very precise regulation of how lipid metabolism can be regulated without affecting anything else, this will be a perfect case for creating a drug.
Yes, sir.
Any
Any
other
other question?
participants?
Sir,
Yeah.
And Ritika, please.
so yeah, can we relate? There is see when there is a knockout, there's a low expression of genes in testes, which was not observed in spleen and brain. So can we relate this to the something like fat related, like there's decrease of fat, decreased fat protection. So can we relate these two?
Uh, okay, the test is gene expression. What we saw mainly they are pseudogene and predicted gene, and we don't really know what those genes does actually, whether they make any protein or
Okay,
not. So we cannot really correlate that directly. Of course, we check the fertility of these mouse, and that is not impacted.
okay.
Yeah.
Sir, one more thing. So if we could, like, did you do the gene expression check in liver? Because when we do lipid metabolism and kind of thing, we need to check those tissues also, right? Like adipose tissue,
Oh,
gene
exactly,
expression.
exactly. So that is something which I'm doing at the moment, that analyzing the RNA expression data from the liver, the fat, and also the hypothalamus, which is the center which regulate energy metabolism in
Huh.
the body. So these things we are doing at the moment.
Okay, sir.
I hope we have some interesting result from there.
Okay.
Yeah. Oh, that's a
Yes.
very good point.
So I think no one
Thank
have
you.
more questions. Sakil, Srilata,
So
Manisha,
just
Chavi,
one
Vijay.
thing I wanted to say, as Dr. Bhatt mentioned, that the coronaviruses are also RNA viruses, and they are very important at the moment. And one thing you may not know, but the coronavirus genome, it start all the time with adenosine as the first nucleotide.
Okay.
And you can imagine that it might have an important role why it keep adenosine as the first nucleotide, because this is something modified. I mean, there are two different things. That the first nucleotide is not only important for this gene regulation, but it's also very important for escaping from the immune system recognition, because that's why the sugar of the first nucleotide is always modified. Because if it is not modified, the the host cells consider this as a foreign RNA, and then they immediately start attacking the RNA. So
Yes.
to protect that, coronavirus bring its own methylase to modify the sugar of the adenosine. But does it also modify the base? And that can provide it additional stability, right?
Right.
And sir, one more question is sir that in SARS-CoV-2, the protein, the
Mhm.
glycoproteins and protein glycoproteins which we target the most, and which and generally sir, this acts as a ligand for the receptors, which are
Yeah,
present
yeah.
in our lungs. And yeah, so sir, if we just target those N genes which
Mhm.
have sir adenosine rich regions, and if we not only just methylates them, we also deacetylase them, then it may helps us to reduce those expression of those N proteins, and that will reduce the viability of those virus,
No,
I think.
exactly. I mean, there are multiple ways the virus can be regulated, and number of people working on different aspects. What I wanted to say, that which is more relevant for this, what we are studying is that since the coronavirus genome always start with A, does it have this methylation? And if this methylation can help the virus to be more stable in the cells, so that can be another drug target to destabilize the coronavirus genome itself, you know, and that can reduce the infectivity.
Okay, sir.
So I think one more aspect
Yeah.
to be highlighted, that the role of antisense RNA, that you have worked a lot. I was just reading your CV.
Yeah.
You started with the pi RNA and antisense
Mhm.
RNA, and a lot of work you have done and published paper in the
Yeah.
Cell also. So can you give a
Mhm.
just a brief idea about the pi RNA antisense RNA? Because just if you'll see in the transgenics, either plant or animals, a lot of these
Yeah.
antisense are not making sense. So,
Yeah, yeah. Okay. So I mean, I did not put a lot of different topic because I thought it may become confusing if I kind of include many different topics. But of course, I can definitely give a brief idea what the pi RNA are and how, what they do actually in the cells.
yeah,
So
we
pi
are
RNA,
very much interested.
so the pi RNA are another class of small RNA, and they are very very similar to the microRNA. If I guess most of you knows about microRNA and how they regulate gene expression, right? So piRNA are specific small RNA which are designed to control the transposon activity in the genome. And the transposon activity is very high in the germ cells. And you know that the germ cells is the carrier of genomic information from one generation to another generation. So it's of a very high interest for the cells to protect its genome in the best way, so that it can transmit the genome information from one generation to another one in a best way, in a more clean way. And that's where the piRNA are important. And what they do is that the piRNA, they bind to argonaute family protein, and then they guide these proteins to recognize the transposon genes, transposon mRNA in the cytoplasm. Since most of the argonaute protein, they have the endonuclease activity. So once they are recognized, the piRNA actually guided cleavage happen, and those RNA are completely degraded in the cytoplasm. So no transposon mRNA can be translated into protein and start jumping in the genome. The second regulation which piRNA provide is they bind to some protein which are nuclear in nature. So they basically translocate to nucleus once they bind to the piRNA. And then they recruit many DNA and histone modification enzyme to really suppress transposon gene expression at a transcriptional level. So the piRNA provide two different way of regulating transposon. The immediate threat is taken care by degrading the mRNA in the cytoplasm. And then long-term protection is provided by modifying the DNA of the transposon promoter, therefore repressing their transcription forever, at least in that generation. So that is how the basic mechanism works for the piRNA mediated gene regulation.
So, sir, generally the piRNA generally used to target the retrotransposons only, or DNA transposons also?
Well, this is mainly the retrotransposon because this is a RNA-based mechanism. So they need to interact with the RNA, right?
Okay, sir.
And in higher organism, basically most of the DNA transposons, they are not active. It's
Yes, sir.
mostly the retrotransposon which get activated during the germ cell formation.
Okay, sir.
And this happened because of the genomic reprogramming which we need to have to kind of create the germ cells. And because of this transposon get activated. So the piRNA then take care of those active transposon during this differentiation process.
Okay, sir. Got it.
So I think participants, anyone want to ask any more question, or we should just end the session by vote of thanks?
You can also ask question about non-coding RNA. I mean, I do
that's very important, non-coding RNA. Because since you have you did a marvelous work, Arindam, just a lot of noise is there.
Yes, sir. I have also.
Okay. So since you did a lot of work over the RNA, and you know better what may be the future of RNA and where current researchers should point out, they should read more, and what is the best out of this RNA part, because all these students are going to do some kind of similar research in future, and they are preparing for CSIR NET examination, and hopefully they will qualify also. So
no, for sure, if they're working hard, they will qualify.
sure.
That's the key, right?
Yeah.
So non-coding RNA, I would say, I mean, the piRNA field is more or less saturated. That was one of the reasons why I moved out of the field and moved to this new area, RNA modification. RNA modification definitely is one of the most exciting areas. It's kind of important for almost every regulation you can imagine at the RNA level. So I would say looking position in RNA modification area will be very interesting. And I just give like a brief introduction of what RNA modifications are, but there are a number of RNA modification and they regulate the gene in n number of ways you can imagine. So this is something is going to stay for a long time. Regarding the non-coding RNA, okay, I would say microRNA and piRNA, they are not so much exciting anymore because most of the things are studied, and only most difficult questions like structural biology questions remain unanswered. Most of the other questions are answered already. Long non-coding RNA, since it is also very diverse, I would say there is still a scope to work in that area. And combining the non-coding RNA with, let's say, RNA modification, since non-coding RNA are kind of dysregulated in many different kind of cancer, it can be still very interesting area to work in.
So I think there is enough question we have taken. Not to waste a single. Anyone is there to ask question?
Yes, sir. So I'm
Please.
Sapna.
Yeah.
Okay, Sapna, please.
So I asked the question: why the product of COVID-19 mRNA vaccine cannot be presented by the MHC?
Pardon?
Well, I don't know that I'm not in the immunology.
mRNA.
She is asking about
Well,
mRNA.
mRNA, but you do not present mRNA to the cells, right? You basically present the protein. And that is completely an immunology question, which unfortunately I don't work in and I cannot really answer.
Okay, I will help you. I am immunologist and everything. I'm dealing with all part of biotechnology basically. And I will tell you, Sapna, definitely I am going to answer this question.
Sir, I just have a question, sir, that generally I'm trying to acquire my PhD from
yes.
abroad, sir. And generally I'm looking at in the University of Geneva recently, and I just want to my research field area of interest is sir structural biology and developmental biology.
Uh-huh.
So if you can provide me sir any aims and scopes related to these two field of interest in which I can apply the molecular biology level to structural biology and generally in developmental biology, and which are the latest areas which are right now covering if, the molecular biology in aspect to the developmental biology. So it will be very
Well,
beneficial for
developmental
me.
biology, yeah, the developmental biology, I would say there is more work done on understanding gene expression again, but more on single cell level. There's single cell sequencing is something being used more regularly, but the developmental biology question remain the same. I mean, how one cell become a whole organism. There is not something changed in that one, and we still need to understand a lot. I would say more focus is on understanding the transcriptional gene regulation during development, and that is something being actively investigated by different people in our department and also in other places. For developmental biology, I would say, yeah, Geneva University, there are few groups, but EMBL you could think of. EMBL is one of the best places for developmental biology.
Okay, sir.
Yeah.
Okay.
And also structural biology, because the EMBL, the good thing is that they have a specific unit for each a topic. Structural biology they have one, for developmental biology they have one, so you can find all sorts of topic there. And also the researcher there are very high quality,
Okay, sir.
and they have this international PhD program which you can apply for, and if you get selected, I think that's one of the best places to do PhD in Europe.
Okay, sir. And sir, Pillai's group that I was looking for in University of Geneva, that the Pillai's
Mhm.
group generally used to sir focus on the molecular biology only, on the gene regulation in transcripts also. And I
No,
think
actually, in our lab, we do both molecular biology, mouse genetics, and some structural biology. So we combine multiple aspects together.
They require a GRE
okay,
TOEFL
sir.
also
No,
to submit the
in
application.
Europe, generally, you don't, I don't think
No,
in
sir. In
Europe it is.
generally, sir, in yeah. No, sir. In Europe, sir, GR is exempted, sir. Only IELTS is generally recommend
Okay.
for the admission
No, because
purpose.
I never applied, so I don't really know if the GRE TOEFL is required. But as far as I remember, it's not really essential, because most of the European countries they have different language. So yeah.
Okay.
Okay, sir.
So,
Excuse me, sir.
Venkatesh, please.
Sir, how many types of rDNA technology, sir?
So what?
rRNA, how many types of RNA?
How many types of rRNA?
RNA, ribonucleic acid.
How many types?
Uh,
Well, you can categorize basically in two types: the protein coding and the non-coding. Of course, in the non-coding, there are multiple categories like microRNA, small interfering RNA, piRNA, then you have tRNA, ribosomal RNA. I mean, I can go on telling you, but this is, yeah.
okay, sir. Thank you.
Thank you, Venkatesh. Anyone else? Any other audience participants? Sri Lata, Sakil, Rathi, Manisha, Vijaya, Ayana.
Sir, generally, right now, just the latest technique has been revived, sir, for CRISPR-Cas9 genome
Yeah.
editing. And yeah, I just read a paper,
That's
and
a very,
I
very
don't
good.
know in brief about it. Say, sir, yeah, and it's sir before sir 1 to 2 months I shared it to Shailendra sir. So I think it's a in they are using sir different kind of viruses along for the process of transfection in addition to that genome CRISPR-Cas9 in addition genome editing also. Can you please, sir, give me a brief about it, sir?
Can, can, can,
For
can
the
you
latest technology which
can
has
you
been.
repeat your question again, because I think I got mixed up with many different things you asked.
Yeah, sir. Actually, sir, there is a just a right now just a latest publication regarding CRISPR-Cas9, and a Nobel
Uh-huh.
Prize has been
We want to know about the CRISPR-Cas9
given to a
technology because
scientist,
this is a
two
kind
to
of
three scientists briefly.
very
I'll
similar
No,
to
but what is
remember
your
it.
question?
restriction
Yeah,
enzyme.
sir. The question is, sir, what is that particular mechanism of action? Yes, sir. Mechanism action is what about what of that the recent advancement of that. CRISPR-Cas9 and
So
genome editing.
your question is how
No, sir.
CRISPR-Cas9
How
work, or
that the paper, the Nobel Prize which was given to those scientists, what they have actually,
Mhm.
what modifications they have done to that CRISPR-Cas9 genome editing? Yes, sir. Because they have generally,
Okay.
yeah,
So okay, if I got your question correctly, what you're asking is what was the basic discovery which led to the Nobel
yeah, sir.
Prize? Yeah. Okay. So how CRISPR-Cas9 work? So in the bacterial genome, we have this CRISPR, the Cas9 proteins, and
Yes,
then you
yes.
have the CRISPR array, which are basically the pieces of viral DNA, which is integrated in the bacterial genome. And this
Yes, sir.
produces a long non-coding RNA, which are then chopped into small pieces, which then bind to the Cas9 protein. And this then act as a defense mechanism. When the new virus come, it can quickly recognize that virus genome and it can cut that. Yeah.
Okay, sir.
So what
Right.
these the scientists which got Nobel Prize, what they did is, since the CRISPR RNA is present into two different pieces, one which bind to the Cas9 protein, and then one which recognize the viral genome. Yeah. So the
Okay.
part which bind to the Cas9 protein is always constant. This is the same RNA all the time. But the part which bind to the viral genome is always different. And that's what being used as a tool to edit genome in the mammals and many other organisms. So what
Okay.
they did actually, they fused these two RNA together, and they showed that by fusing these RNA, you can just use one RNA, one protein, and then combine them, and you can edit basically anything. So the variable part you can change with whatever you want, and the constant part will be there all the time. And by combining this one, you can basically edit anything you want.
Okay, sir.
Did I answer your
So
question?
we can, yes, sir. So we can generally that eliminate those lethal genes also, sir, and we can also use we can also can eliminate by using those genes which can be for the lethal as
Yeah,
well.
yeah, basically, they provided the tool that if you just change this variable region to anything which you are interested in, you can target that gene.
Okay, sir. So, sir, actually,
Yeah.
generally, in right now, in if I talked about in Rajasthan, I'm from Rajasthan, sir, and generally from the past 2 to 3 months, the cases of psoriasis uh, is getting so much high, sir. Everyone,
Mhm.
even out of, sir, in every district, sir, out of 100, 20 to 30 psoriasis patients are getting highlighted. So, if we used
Mhm.
to, it's called psoriasis genes are, sir, generally, sir, recessive, and they used to express in in 2 to 3 generation in a skipped manner. So
Mhm.
if we can eliminate those genes like PSORA1 and PSOAR2 genes,
Mhm.
generally, with the help of this CRISPR-Cas9 genome editing,
Mhm.
we can surely eliminate this psoriasis disease in
No, no,
very
of
fast.
course. I mean, of course. I mean, this CRISPR-Cas9 is so versatile. You can basically target any gene. You just need to change the variable region to your interesting gene, right? And then you can target it, because this protein doesn't care which gene it binds. It just need to land on DNA, and that happened because of the base pair between this variable region and the target region. So just by changing variable region to any target, you can basically target Cas9 wherever you want.
Yes, sir. Okay, sir.
So there is one question from Sapna.
Mhm.
Exonuclease domain containing some enhanced region like
Yeah,
maybe 2 pi RNA
yeah,
biogenesis.
yeah.
So they are going to interact
Uh-huh.
with what?
The EXD1.
Exonuclease domain that containing one enhances maybe 2
Yeah.
pi RNA biogenesis.
So this,
They are going to interact
yeah,
with.
so this EXT1 actually, the EXT1 interact with another protein called Tudor domain containing 12.
Yes,
And these two proteins together actually help in biogenesis of nuclear pi RNA.
right,
So
right.
once they associate with the nuclear PV protein, they help in producing nuclear pi RNA and loading them onto the PV protein, the MeV2. So if you don't have these two genes, basically nuclear PV protein cannot bind to pi RNA, and then it cannot function.
Okay. Right. So I think
Yeah.
that's enough question we have taken. So all the participants are requested. If you have any question, you can WhatsApp me later on. All right. We will try to answer you as soon as possible. And I will also going to share you the video on my YouTube channel. So from there you can get. But PPT will not be provided because this is the research work of Dr. Saab. So
Yeah. Yeah.
uh, so that cannot be provided because of copyright violation and a lot of things.
No, because
Because
many of the things are not published yet. So I would say that yeah, better not to put on YouTube.
public domain. Recording can be given as a
Yeah.
part of
Yeah.
lecture series, that on the subscription.
Yeah. Yeah. Yeah.
So,
No, that's not a
uh,
problem. That's not a problem.
thank you so much, Dr. Saab, for giving so much fruitful knowledge, so much advanced knowledge about the RNA. We all were reading so much, but the concept was not so much clear. So that's when you said to me that I'm ready to give a talk on Dharani. I was so much happy that because last time I was having a very bad experience that
Uh-huh.
when from USA, one of my friend was ready to give the seminar and webinar, and then I invited and shared on the Facebook, LinkedIn. More than 200 people registered, and when the webinar was delivered, only 15,
Mhm.
10 to 12 people were there. So that was free of cost. That's why I thought people should realize,
Oh,
right,
I mean,
the importance of the
it
things.
is
If
a
they are
loss of
paying,
those people who,
huh,
yeah,
so
no, it
that's
is loss of
why,
people, those who are not attending. I am spending
huh,
my time to
managing all the
give what
things,
I know,
right?
right?
So that's why people in India, and even I have observed that the students are not very serious. I have selected only 10 students out of 90 student
Mhm.
via the scholarship test, because I know a lot of garbage students are there. They don't know the ABC and they are going to give me a kickback in the results. So everyone is asking the results. So that's why I selected very, and you have seen what kind of talent are they having, and
Yeah,
our all the students are
yeah.
meritorious. That's why I have given a chance to interact with you. They should know where the world is going on, where the latest technology is going on, because only
Yeah.
I am telling here and they are listening me just, and I do not want to make this limitation that they should only listen to me. And that's why in the year I'm giving more than 12 to 15 seminars from worldwide. And in the January 17, and one of the professor from USA, Dr. Very food technologist, I forget his name, I think. Mm. He was my supervisor, and he was just be part of my PhD sessions. So I invited Kalidas Setti. Yes, I remember Professor Kalidas Setti from USA. He is going to deliver a very good seminar over the food sciences and transgenic nutraceutical. A lot of very good experiences. He is the editor of Food Biotechnology Journal in
Mhm.
the Taylor Francis. So I just requested for my students. So I am making this arrangement so make them enrich. And that's why I am very thankful to you. In
Yeah.
very short time, you accepted all my invitation and given us such a wonderful, informative
No,
session, such a
this
knowledgeable,
kind of
and
it
it's a
would be
very
fine for me
rare.
if I can help people learn something. Then
Yes,
I'm much more happy. If it help
yes.
to learn even few things, I think that's more than enough for me.
Yes, and all my students, I hope I will get something more than I have given. they they were getting knowledge of the pi RNA and mRNA, silencing RNA. I was just telling them as a part of syllabus, so I was not dealing them with much research. But you have given a wonderful explanation of all this via the your experimental works, and a lot of technology related to the lipid analysis and RNA analysis, and how the SAM is going to be activating, adding methyl group over the adenine, and beautiful knowledge about the this corona RNA viruses, that it is going to start with the A. And definitely my students in the future, when they will go for the research, they will think about it, and they will not going to jump in that field that is already saturated, because a lot of students don't know in India what is the current field where they should jump out. They are just taking a JRF as a their job. Means they are employed. If they are
Yeah.
qualified JRF, they are
Well,
not taking
but
seriously.
you know, but you know, if they are for a job, then you are unemployed for 5 years.
Yes. That's why I want to give a message via this platform, that all students should take very seriously this research career. This is
Yeah.
very informative, this is very interesting, and then easily you can qualify the CSR. CSR is not a kind of big hurdle, it's not a Mount Everest that you
No,
have to jump.
definitely not.
The
AR is
consistent.
not the most important thing. The most important thing that you have to like this field, otherwise you cannot survive.
Yes, because if you have interest, automatically you are going to gain every day, and you have automatic motivation behind learning a lot of things, because you have to cover a lot of books from the gene 12 and molecular biology, and then developmental biology. A lot of book you have to cover. Star in our old time, we can remember in the BHU, we were studying the Strayer, Leninger, and all that standard books,
Yeah.
not lesser
Yeah.
than that. No Indian books except BD Singh book. That was the only one Indian book.
There was no choice in that
And that
one.
was no choice, because Bhojwani was not so much clear and updated. That's why we have to leave that. And over the molecular biology, except Dr. Tripathi was enhancing, enriching so much knowledge, that we have to follow at least 4 to 5 books to get his one lecture only. So I
Yeah.
request all of my students not to jump behind those teachers in their preparation. जैसे कि कुछ लोगों ने जेआरएफ निकालते ही वो टीचर बन जाते हैं कि भाई आज हम टीचिंग करेंगे और जेआरएफ निकालेंगे सबका. तो दैट्स व्हाट आई हैव सीन दैट प्रैक्टिस इज गोइंग ऑन, कि चाट पकौड़े खा रहे हैं लोग नॉलेज में. तो इस तरह से रिसर्च नहीं हो पाएगी. आप लोगों को थोड़ा सा अच्छे टीचर्स को ज्वाइन करना चाहिए, और देखना चाहिए कि रिसर्च वाले बंदे कहाँ पे हैं. तो अगर आप चाहते हैं, जैसे प्लेटफॉर्म आजकल चल रहे हैं, कुछ नाम मैं नहीं लेना चाहूँगा, कि वो बच्चे एमएससी का एबीसीडी नहीं जानते हैं, और दे आर टीचिंग टू जेआरएफ पीपल. So that's what kind of practice is going to spoil the whole research ethics, है ना.
Yeah.
So thank you,
Well,
Dr. Saab,
research.
once again. If you have
Yeah.
any message, please give a beautiful message to our MSc students, because they are not only the future scientists, they are going to handle the career of others also. So if they will learn from us the right education, then they can guide the same fraternity to other students also.
I mean, I would say it's better to take your time and decide for research, because this is a long game. If you are finding yourself not very much interested and motivated, don't come into research.
Right. Okay. Thanks once again, Dr. Saab,
Thank you, sir.
for giving
Yeah,
so
thank
much
you very
Thank
much.
you
beautiful
so
explanation
much,
of
sir.
everything. So I will be in touch of you, and my students will also will take guidance from time to time. Thank you so much
Yeah,
for your contribution. Thank you, Dr. Saab.
thank you. Okay. Bye bye.
Thank you so much.